Sperm cryopreservation currently uses two types of carriers, cryotubes and straw tubes. Remove the cryotube from liquid nitrogen using forceps. Exercise caution to avoid skin injury. Wipe off the frost from the cryotube surface with sterile tissue. Unscrew the cap, ensuring no liquid nitrogen remains, then securely re-tighten the cap. Place the cryotube into a 37 degrees Celsius water bath on a foam board. Gently rock the foam board back and forth, ensuring the cryotube is fully immersed and in contact with the warm water. Set a timer for 10 minutes. After 10 minutes, remove the cryotube from the water bath and wipe off any residual water droplets with sterile tissue. Carefully unscrew the cap of the cryotube and use a micropipette to transfer 20 microliters of sperm into a 90 microliter activation dish on a 37 degrees Celsius heated plate. We will now introduce the thawing process for the straw tube. Remove the straw tube from liquid nitrogen using forceps. Wipe off the frost from the straw tube surface with a sterile tissue. Place the straw tube into a 37 degrees Celsius water bath on a foam board for 10 minutes. After 10 minutes, remove the straw tube from the water bath. Wipe off any water droplets from the straw tube surface using sterile tissue. Then use sterile scissors to cut off both ends of the straw containing the sperm. Here is the straw tube for your reference. Next, let's activate the sperm by transferring it into the activation dish. Connect the straw tube to the micropipette and gently expel 10 microliters of sperm into the 90 microliter activation dish by pipetting back and forth. Ensure this step is performed on a 37 degrees Celsius heated plate. The following steps are the same for both the straw tube and cryotube. Place the sperm dish into a 37 degrees Celsius 5% carbon dioxide incubator for 30 minutes of activation. After 30 minutes, remove the sperm dish from the incubator and observe the sperm concentration and motility under the microscope. Using a micropipette, transfer 20 microliters of high motility sperm from the activation dish and add it to a HTF and GS egg droplet for fertilization. Ensure this step is done on a 37 degrees Celsius heated plate. Return the dish to the incubator for about 4 hours for fertilization to occur. After fertilization, transfer the well-formed full one-cell embryos into fresh M16 medium. Leave any impurities and fragmented eggs in the original droplet. Repeat this procedure 2 to 3 times to ensure thorough cleaning. After approximately 16 hours of culture, select the well-formed 2-cell embryos. Wash them and transfer them into fresh M16 medium for transplantation.